In this video, we are going to learn about NMR and electron spin resonance spectroscopy, parts, working, and applications. So, nuclear magnetic resonance or NMR and electron spin resonance ESR spectroscopy are powerful analytical techniques used to study the physical, chemical, and biological properties of molecules. Here is an overview of the parts, working, and applications of NMR and electron spin resonance spectroscopy. Nuclear magnetic resonance spectroscopy. The main parts of an NMR spectrometer include a magnet, a sample holder, a radio frequency transmitter and receiver, and a computer system for data acquisition and analysis. NMR spectroscopy involves the measurement of the magnetic properties of atomic nuclei in a magnetic field. When a molecule is placed in a magnetic field, its atomic nuclei become magnetized and can absorb and emit radio frequency energies at a specific frequency that is depend on the strength of the magnetic field and the chemical environment of the nucleus. By measuring the absorption and emission of radio frequency energy, information about the molecular structure, dynamics, and interactions can be obtained. So, what is happening here? NMR spectroscopy involves the measurement of the magnetic properties of atomic nuclei in a magnetic field. We will be putting an atomic nuclei in a magnetic field and then measuring the magnetic properties of that particular atomic nuclei. When a molecule is placed in a magnetic field, its atomic nuclei become magnetized and can absorb and emit radio frequency energy at a specific frequency that is dependent on the strength of the magnetic field and the chemical environment of the nucleus. By measuring the absorption and emission of radio frequency energy, information about the molecular structure, dynamics, and interactions can be obtained. NMR spectroscopy is widely used in chemistry, biology, and medicine for structural elucidation, identification of unknown compounds, determination of reaction mechanisms, and investigation of protein structure and dynamics. Next is the electron spin resonance spectroscopy. The main parts of an ESR spectrometer include a magnet, a microwave source, a sample holder, and a computer system for data acquisition and analysis. ESR spectroscopy involves the measurement of the magnetic properties of unpaired electrons in a magnetic field. So, what is the working principle? ESR microscopy involves the measurement of the magnetic properties of unpaired electrons, in a magnetic field. When a molecule with an unpaired electrons is placed in a magnetic field, the electrons become magnetized and can absorb and emit microwave energy at a specific frequency that is dependent on the strength of the magnetic field and the chemical environment of the electrons. By measuring the absorption and emission of microwave energy, information about the molecular structure, dynamics, and interactions can be obtained. So, what are the applications of electron spin resonance microscopy? ESR microscope ESR spectroscopy is used in chemistry, biology, and physics for studying free radicals, paramagnetic species, and magnetic materials. It is also used in medicine for diagnosis and treatment of diseases such as cancer and cardiovascular diseases. What is the importance of NMR and ESR spectroscopy? These techniques are important analytical techniques that have revolutionized many fields of science. They provide valuable information about the structure, dynamics, and interactions of molecules, which is essential for understanding the physical, chemical, and biological properties of molecules. They are also used in drug development, material science, and environmental analysis, along with other fields. So, structure determination using X-ray diffraction crystallography. Next topic is the structure determination using X-ray diffraction crystallography. X-ray diffraction crystallography is a technique used for determining the three-dimensional structure of molecules, including proteins, nucleic acids, and small molecules. There is an overview of the parts, working applications, and importance of X-ray diffraction crystallography. Which are the parts? The main parts of an X-ray diffraction crystallography system include an X-ray source, a monochromator, a collimator, a sample holder, a detector, and a computer system for data acquisition and analysis. So let's learn about the working. X-ray diffraction crystallography involves the use of X-rays to study the arrangement of atoms in a crystal. When X-rays are directed into a crystal, they are diffracted by the atoms in the crystal, producing a diffraction pattern. The diffraction pattern is captured by a detector, and the data is processed using mathematical algorithms to obtain a three-dimensional map of the electrical density distribution in the crystal. This information can be used to determine the positions of atoms in the crystal, and thus the three-dimensional structure of the molecule can be easily recognized. So, which are the applications? X-ray diffraction crystallography is used in chemistry, biochemistry, and structural biology for the determination of the three-dimensional structures of molecules. It is particularly useful for the study of proteins, which are difficult to study using other techniques due to their complexity and size. The structural information contained from X-ray crystallography can be used for drug discovery, understanding enzyme mechanisms, and the designing new molecules with specific properties. So, what is the importance of X-ray diffraction crystallography? It is a critical technique for the study of molecular structure and has been used to determine the structure of many important biomolecules, including DNA, insulin, and hemoglobin. It has also played a key role in the development of new drugs for diseases such as cancer and HIV and Alzheimer's disease. The technique is highly precise and accurate and has advanced our understanding of the structure function relationships of molecules in biological systems. Next, we are going to learn about a major topic that is nothing else. Circular dichorism, parts, types, working and applications and importance. Circular dichorism is a technique used to study the optical properties of chiral molecules. Here is an overview of the parts, types, working, applications and importance of circular dichorism. What is the importance of circular dichorism? That is used to study the optical properties of chiral molecules. The main parts of a circular dichorism spectrophotometer include a light source, a monochromata, a polarizer, a sample holder, a detector, and a computer system for data acquisition and analysis. So, there are two main types of circular dichorism: electronic circular dichorism or ECD, and the vibrational circular dichorism or VCD. So, ECD is used to study the electronic transitions of chiral molecules, while VCD is used to study the vibrational transitions. Wait a minute. There are two main types of electrical dichorisms. First is the electronic circular dichorism. Next one is the vibrational circular dichorism. Electronic circular dichorism is used to study the electronic transitions of chiral molecules, while vibrational molecular dichorism is used to study the vibrational transitions of molecules, chiral molecules. So, working of circular dichorism. Circular dichorism works by passing polarized light through a sample containing a chiral molecule. As the light passes through the sample, it interacts differentially with the left and the right-handed enantiomers of the molecules, leading to differences in absorbance. The difference in absorbance between left and right circularly polarized light is called circular dichorism. What is circular dichorism? The difference in absorbance between left and right circularly polarized light is called circular dichorism. The CD signal is measured using a detector and the data is processed using mathematical algorithms to obtain information about the structure and properties of the chiral molecules. Now let's learn about the application. Circular dichorism is used in chemistry, biochemistry and structural biology to study the properties of chiral molecules including proteins, nucleic acids and small molecules. Structural biology to study the properties of chiral molecules including proteins, nucleic acids and small molecules. It is particularly useful for the study of protein structure and folding as well as the interactions between proteins and ligands of other molecules. It can also be used to study conformational changes in DNA and RNA. What is the importance of circular dichorism? Circular dichroism is a highly sensitive technique for studying the optical properties of chiral molecules and is an important tool for the structural characterization of biological molecules. It can provide information about the secondary and tertiary structure of proteins as well as the conformational changes that occur upon ligand binding or other interactions. It can also be used to study the effects of mutations or other modifications on protein structure and function. First of all, let's learn about the working once again. Circular dichroism works by passing polarized light through a sample containing a chiral molecule. As the light passes through the sample, it interacts differentially with the left and right-handed enantiomers of the molecule, leading to differences in absorbance. The difference in absorbance between left and right circularly polarized light is called circular dichroism. The CD signal is measured using a detector and the data is processed using mathematical algorithms to obtain information about the structure and properties of the chiral molecules. So, which are the parts? As mentioned earlier, there are two main types, which are the electronic circular dichroism and the vibrational circular dichroism. So, circular dichroism works by passing polarized light through a sample containing a chiral molecule. As light passes through the sample, it interacts differently with the left and right-handed enantiomers of the molecule, leading to differences in absorbance. The difference in absorbance between left and right circularly polarized light is called circular dichroism. The CD signal is measured using a detector and the data is processed using mathematical algorithms to obtain information about the structure and properties of the chiral molecule. Now let's move on to the next topic, that is nothing else but the surface plasma resonance technique. Surface plasma resonance technique and its principal parts, working and applications. Surface plasma resonance. Surface plasma resonance is a powerful analytical technique used to study the interactions between biomolecules such as proteins, nucleic acids and carbohydrates with high specificity and sensitivity. Here is an overview of the principles, parts, working and applications of SPR. What is SPR? It is a powerful analytical technique used to study the interactions between biomolecules. Here we are studying the interactions between biomolecules such as proteins, nucleic acids and carbohydrates with high specificity and sensitivity. So how to do this and what's the principle behind that? The principle of surface plasma resonance is based on the interaction between light and the electrons at the interface between a metal film and sample solution. So what is happening here? There is an interaction between light and the electrons at the interface between a metal film and a sample solution. When polarized light is incident on a metal surface, the electrons of the metal film oscillate collectively. When polarized light is incident on the metal surface, we are colliding a polarized light on the metal surface of a particular particular substance. When polarized light is incident on the metal surface, the electrons of the metal film oscillate collectively, forming a surface plasma wave. This surface plasma wave can be detected as a decrease in the intensity of the reflected light, which is highly sensitive to changes in the refractive index of the sample solution. The main parts of a surface plasma resonance instrument include a light source, a prism, a metal film, a flow cell for sample injection, a detector, and a computer system for data acquisition and analysis. Working. A sample solution containing a biomolecule of interest is injected into the flow cell, which contains the metal film coated with a thin layer of receptor molecules that can specifically bind to the biomolecule. So, first of all, a sample solution containing the biomolecule of interest is injected to a flow cell, which contain a metal film coated with a thin layer of receptor molecule that can specifically bind to the biomolecule. As the biomolecule binds to the receptor molecule on the metal surface, the refractive index of the sample solution changes, leading to a shift in the angle of the reflected light.
The shift in the angle of the reflected light is proportional to the amount of biomolecule binding and can be used to determine the binding affinity and kinetics of the interaction. Once again, I'm going to read. A sample solution containing the biomolecules of interest is injected into the flow cell, which contains the metal film coated with a thin layer of receptor molecules that can specifically bind to the biomolecule. As the biomolecule binds to the receptor molecules on the metal surface, the refractive index of the sample solution changes, leading to a shift in the angle of the reflected light. First, we are going to apply the solution, where it is injected into a flow cell, and that flow cell contains a metal film coated with a thin layer of receptor molecule that can specifically bind to the biomolecule. So, into a metal film coated with a thin layer of receptor molecule, we are applying the biomolecule sample into a flow cell. As the biomolecules bind to the receptor which is present on the metal film, the refractive index of the sample solution changes because there is happening a binding. So its refractive index changes, leading to the shift in the angle of the reflected light. The shift in the angle of the reflected light is proportional to the amount of biomolecule binding, and it can be used to determine the binding affinity and kinetics of the interaction. So there will be a suitable receptor which is coated on a metal plate, and flow cell will be there, and we are injecting the corresponding sample into that. There will be a binding occurring between the receptor molecule and the biomolecule. Then the refractive index of the sample solution will change accordingly. So SPR is widely used in the fields of drug discovery, biophysics, and proteomics to study protein, DNA, and protein ligand interactions. It can be used to screen small molecule libraries for potential drug candidates. It can be used to screen small molecule libraries for potential drug candidates to determine the binding affinity and kinetics of protein-protein interactions and to study the conformational changes that occur upon ligand binding. It can also be used to study the specificity of antibody-antigen interactions and to detect biomarkers in clinical samples. What is the importance of SPR? SPR is a label-free, real-time, and highly sensitive technique for studying the interactions between biomolecules, making it a valuable tool for drug discovery and structural biology. It can provide information about the binding kinetics, affinity, and specificity of biomolecular interactions, and can be used to study conformational changes in proteins and nucleic acids. Its high sensitivity and specificity make it particularly useful for the detection of low abundance analytes in complex biological samples. Next, electrophysiological methods. We are going to learn about the electrophysiological methods like single neutron recording and its principle along with parts, working, and applications. So, first electrophysiological method is the single neutron recording. What is this particular single neutron recording? This is an electrophysiological technique used to study the electrical activity of individual neurons. Here is an overview of the principle, parts, working, and applications of single neuron recording. The principle of single neuron recording is based on the fact that neurons generate electrical signals called action potentials when they are activated. By inserting a microelectrode into the extracellular space near a neuron, it is possible to detect the changes in electrical potential associated with a neuron's activity. The main parts of a single neuron recording setup include a microelectrode, an amplifier, a signal processor, and a recording device, which are the main parts. Definitely we're going to need a microelectrode because that is something which captures the action potential. And the action potential of one single neuron is very small, so we need an amplifier for that purpose to amplify that signal, and a signal processor is needed, and a recording device or a screen or something which an electrical method to show that result. The microelectrode is a thin insulated wire that is inserted into the extracellular space near the neuron. The amplifier amplifies the electrical signal detected by the microelectrode. The signal processor filters and digitizes the amplified signal. The recording device stores the digitized signal for later analysis. How this works? The microelectrode is inserted into the extracellular space near a neuron using a micromanipulator is using here. The electrical signals generated by the neuron are detected by the microelectrode and amplified by the amplifier. The amplifier signal is then filtered and digitized by the signal processor. The digitized signal is recorded by the recording device for later analysis. So here is a micromanipulator is a very important one, because which helps to insert the microelectrode into the extracellular space near a neuron. So what is the application of a single neuron recording? It is used in neuroscience research to study the electrical activity of individual neurons. It can be used to study the firing properties of neurons in response to different stimuli, to identify the types of neurons present in a given region of the brain, and to study the properties of neurons in disease states. Single neuron recording can also be used in clinical settings to identify the location of abnormal electrical activity in the brain, such as epilepsy. It can also be used to evaluate the efficacy of treatments for neurological disorders such as deep brain stimulation. So what is the importance of single neuron recording? It's a powerful tool for studying the electrical activity of individual neurons, providing insight into the neural mechanisms underlying behavior and cognition. It has helped advance our understanding of brain function and the pathophysiology of neurological disorders. Single neuron recording has also led to the development of new treatments for neurological disorders such as deep brain stimulation for Parkinson's disease and epilepsy. Next one is the patch clamp recording, its principle, parts, working and applications and importance. So what is a patch clamp recording? Patch clamp recording is an electrophysiological technique used to study the electrical activity of individual cells, including neurons, muscle cells, etc. It's the electrical activity of individual cells. Earlier we have learned the action potential of one single neuron. That was measured in the earlier case, like the single neuron recording. Here similar case is happening in the patch clamp recording principle. So patch clamp recording is an electrophysiological technique used to study the electrical activity of individual cells, including neurons, muscle cells and other excitable cells. There is an overview of the principal parts, working, application and importance of patch clamp recording. The principle of patch clamp recording is based on the fact that cells generate electrical signals when they are activated. By using a micropipette with a very small opening, it is possible to create a seal between the pipette and the cell membrane, allowing for the measurement of the electrical activity of the cell. The main parts of a patch clamp recording setup include a micropipette, an amplifier, a signal processor, and a recording device. The micropipette is a thin hollow glass tube with a very small opening at the tip. The amplifier amplifies the electrical signal detected by the micropipette. The signal processor filters and digitizes the amplified signal. The recording device stores the digitized signal for later analysis. So how this works? The micropipette is placed near the cell membrane using micromanipulator. A small amount of suction is applied to the pipette, creating a seal between the pipette and the cell membrane. The electrical signals generated by the cell are detected by micropipette and amplified by the amplifier. The amplified signal is then filtered and digitized by the signal processor. The digitized signal is recorded by the recording device for later analysis. So what is the application of this patch clamp recording? Patch clamp recording is used in many areas of research, including neuroscience, cardiology, and pharmacology. In neuroscience, it is used to study the electrical activity of individual neurons, including the properties of ion channel and synapses. In cardiology, it is used to study the electrical activity of cardiac cells and the properties of ion channels that are important in heart function. In pharmacology, it is used to study the effects of drugs on ion channels and other cellular processes. So what is the importance of patch clamp technique? Patch clamp recording is a powerful tool for studying the electrical activity of individual cells, providing insight into the cellular mechanisms underlying behavior and disease. Cellular mechanisms underlying behavior and disease we will be learning. It has led to important discoveries in neuroscience, cardiology, and pharmacology, including the identification of new drug targets and the development of new treatment for neurological and cardiovascular disorders. Patch clamp recording has also led to the development of new technologies such as orthogenetics, optogenetics. Patch clamp recording has also led to the development of new technologies such as optogenetics, which allows researchers to control the activity of specific neurons using light. Overall, patch clamp recording is an important tool for advancing our understanding of cellular function and disease. Next, we are going to learn about ECG, electrocardiography, its principles, parts, working, and applications, and its importance. So, electrocardiography is a non invasive medical test. Electrocardiography, or ECG, is a non invasive medical test used to detect and diagnose heart problems. And the principle behind the electrocardiography, we are learning about the electrocardiography. Electroencephalopathy is coming. We will be learning about it after this one. So, ECG, electrocardiography, works in the principle of ECG is based on the fact that heart generates electrical signals when it contracts. By placing electrodes on the skin, it is possible to measure these electrical signals and produce a visual representation of the heart's activity. The main parts of the ECG machine include electrodes, an amplifier, a signal processor, and a recording device. The electrodes are placed on the skin at various locations on the chest, arms, and legs. The amplifier amplifies the electrical signals detected by these electrodes. The signal processor filters and digitizes the amplified signal, and recording device stores the digitized signal for later analysis. How this works? The electrodes are placed on the skin at specific locations to detect the electrical signals generated by the heart. The electrical signals are then transmitted to the amplifier where they are amplified, and the amplified signals are then filtered and digitized by the signal processor. The digitized signal is recorded by the recording device for later analysis. So, what is the application of ECG? ECG is used to diagnose a variety of heart problems, including arrhythmias, heart attacks, and other cardiac conditions. It is also used to monitor the effects of heart medication, to evaluate the effectiveness of treatment, and to assess the risk of heart disease in individuals with risk factors. ECG is an important diagnostic tool in cardiology and is widely used in hospitals, clinics, and doctors' offices around the world. It is a non invasive and relatively low cost test that can provide valuable information about the function of the health. Function of the heart. ECG can help identify heart problems early on before they become more serious and can help guide treatment decisions. It is also useful in screening for heart disease in individuals with risk factors such as high blood pressure or a family history of heart disease. Overall, ECG is an essential tool for diagnosing and managing heart conditions, improving patient outcomes, and promoting cardiovascular health. Next one is the brain activity recording. Brain activity recording, also known as neurophysiological recording, is the process of measuring and analyzing the electrical or magnetic activity of the brain. This technique is used to study the function and dysfunction of the brain in both research and clinical settings. This is a technique which is also known as neurophysiological recording. Is the process of measuring and analyzing the electrical or magnetic activity of the brain. Here we are measuring or analyzing the magnetic activity of the brain. Brain activity recording, also known as neurophysiological recording, is the process of measuring and analyzing the electrical or magnetic activity of the brain. This technique is used to study the function and dysfunction of the brain in both research and clinical settings. Here are some common methods used for brain activity recording. First is the electroencephalography, EEG. Electroencephalography. EEG is a non-invasive technique that records the electrical activity of the brain using electrodes placed on the scalp. We will be placing the electrodes in the scalp by electroencephalograph. electroencephalograph, EEG, electroencephalograph. Encephalo in the sense, cephalo is the word something which is related to the brain part or the head region. So that is why it is termed as an electroencephalograph. This is a non-invasive technique that records the electrical activity of the brain using electrodes placed on the scalp. It is used to study brain waves and diagnose and monitor conditions such as epilepsy, sleep disorders, and head injuries. Next is the magnetoencephalography. Magnetoencephalography or MEG, MEG. Magnetoencephalography. Magnetoencephalography is a non-invasive technique that records the magnetic fields generated by the electrical activity of the brain using sensors placed outside the head. It is used to study the timing and location of the brain activity and to diagnose and monitor conditions such as epilepsy, brain tumors, and stroke. Next is the functional magnetic resonance imaging. Functional magnetic resonance imaging is a non-invasive imaging technique that measures changes in blood flow to different areas of the brain, which are used as a proxy for brain activity. It is used to study brain function during different tasks and to diagnose and monitor conditions like stroke, brain tumors, and neurodegenerative diseases. Here we can just see or identify the blood flow changes, it measures the changes in blood flow of different areas of the brain during brain activity. The brain is active here. Next is the positron emission tomography, PET scan. Positron emission tomography is a non-invasive imaging technique that uses radioactive tracers to measure metabolic activity in different areas of the brain. It is used to study brain function and metabolism and to diagnose and monitor conditions such as Alzheimer's disease, Parkinson's disease, and brain tumors. Gamma camera is used here. Then, single photon emission computed tomography. Single photon emission computed tomography is a non-invasive imaging technique that uses radioactive tracers to measure blood flow to different areas of the brain. It is used to study brain function and to diagnose and monitor conditions such as epilepsy, stroke, and traumatic brain injury. Overall, brain activity recording is a valuable tool for understanding the working of the brain and for diagnosing and treating neurological disorders. By providing insights into brain function and dysfunction, these techniques can help improve patient outcomes and promote brain health. Now, let's learn about lesions and stimulation of brain. Stimulation of brain. Lesion and stimulation of the brain are two different techniques used to study the functions of the brain. Lesion studies involve damaging or removing a part of the brain to observe the effects on behavior, while stimulation studies involve applying electrical or magnetic currents to specific areas of the brain to observe their effects. Lesion studies can be done through various methods, including surgical removal, chemical injection, or radiation therapy. Lesions can be either intentional or accidental. For example, patients who have suffered a stroke or head injury can have naturally occurring brain lesions that can be studied. Lesion studies can provide valuable insights into the functions of the different brain regions. For instance, if a specific lesion produces a particular behavioral deficit, then it can be concluded that the lesioned area is involved in the process underlying that behavior. It's similar to the experiment of knockout mice where we just remove certain important portions of genes and behavior changes are observed in the knockout mice. We are knocking out particular genes in that mice and learning about the behavior changes happening in that particular mice. So we can just conclude that this particular change is associated with this gene. So that gene is very important. So never disturb that, or it can be used to learn about the mutations or genetic editing that we need to do in certain animals. So that is the case of knockout mice. The same situation is happening in the lesions, brain lesions, as the lesioned area is included in the process underlying that behavior. On the other hand, stimulation studies involve applying a small electrical or magnetic current into a specific region of the brain to observe its effect on behavior. This can be done through various methods, including transcranial magnetic stimulation, and deep brain stimulation. Transcranial, cranial in the sense head, cranium, transcranial magnetic stimulation and deep brain stimulation. So, transcranial magnetic stimulation involves placing a magnetic coil over the scalp, which generates an electrical field that can stimulate the underlying brain tissue. Then, the deep brain stimulation involves surgically implanting electrodes into specific regions of the brain and using an external stimulator to activate them. Both methods can be used to study the effects of stimulating different brain regions on behavior, which are the two types. Transcranial magnetic stimulation, where we just provide a magnetic coil, applying a magnetic coil on the scalp. Next is the deep brain stimulation. Deep brain stimulation is something which just happens by applying surgically implanting electrodes into the specific regions of the brain and using the external stimulator to activate them. These both methods can be used to study the effects of stimulating different brain regions on behavior. Stimulation studies have provided valuable insights into the functions of different brain regions and can also be used to treat various neurological and psychiatric disorders. For instance, TMS has been used to treat depression, anxiety, and chronic pain, while TMS in the sense, what is the full form of TMS? Transcranial stimulation, deep brain stimulation. Transcranial stimulation has been used to treat depression, anxiety, and chronic pain, while deep brain stimulation has been used to treat Parkinson's disease, essential tremor, and epilepsy. Overall, both lesion and stimulation studies are valuable tools for understanding the functions of the brain. While lesion studies can provide insights into the functions of specific brain regions, stimulation studies can be used to manipulate brain activity and study the causal relationship between brain activity and behavior. These techniques have broad applications in both research and clinical settings and continue to provide valuable insights into the workings of the human brain. Next is the pharmacological testing. Brain activity recording. Pharmacological testing of brain activity involves the use of various drugs to study the effects of these compounds on the functioning of the brain. This can be done through different methods of brain activity recording, such as electroencephalograph, functional magnetic resonance imaging, and positron emission tomography (EEG), or electroencephalograph is a non-invasive technique that records the electrical activity of the brain using electrodes placed on the scalp. Electroencephalogram. The electroencephalogram recordings can provide valuable insight into the effects of drugs on brain activity, including changes in brain activity patterns, frequency, and amplitude. For instance, drugs that increase the activity of neurotransmitters like dopamine can lead to changes in the EEG patterns that are associated with increased alertness, arousal, and attention. fMRI is a non-invasive imaging technique that uses magnetic fields to detect changes in blood flow and oxygenation in the brain during brain activity. This technique can provide information on the areas of brain that are activated by different drugs and can help to identify the neural circuits involved in various brain functions. For example, fMRI studies have shown that drugs which increase the release of dopamine in the brain can activate the reward centers and lead to increased feelings of pleasure and motivation. PET scan is another imaging technique that involves the ingestion of a radioactive tracer into the bloodstream, which is then detected by scanner to provide information of brain activity. PET can be used to study the effects of drugs on various neurotransmitter systems in the brain, including dopamine, serotonin, and norepinephrine. For example, PET studies have shown that drugs that block the reuptake of serotonin can increase its levels in the brain and lead to changes in mood and behavior. Then, pharmacological testing of brain activity is important for understanding the effects of drugs on brain functions and behavior. It can help to identify potential therapeutic targets for various neurological and psychiatric disorders, as well as aid in the development of new drugs that target specific neurotransmitter systems, identify potential therapeutic targets for various neurological and psychiatric disorders, as well as aid in the development of new drugs that target specific neurotransmitter systems. However, it is important to note that pharmacological testing of brain activity should always be conducted with caution and under ethical guidelines to ensure the safety and well being of the study participant. PET, MRI, fMRI are once again dealt here. So, PET, positron emission tomography. MRI is the magnetic resonance imaging. fMRI is the functional magnetic resonance imaging. And CT, computed tomography, are all imaging techniques used in the field of medicine to diagnose and study various techniques. Here is an overview of each of this technique. PET scan. PET scan uses a radioactive tracer that is injected into the body, and as it decays, it releases positrons that interact with electrons in the body, creating gamma rays that can be detected by a gamma camera or a scanner. What is happening here? Positrons are emitted and they interact with the electrons in the body, and this will eventually create gamma rays that can be detected by a scanner. The scanner creates a 3D image of the body based on the gamma rays detected. The PET scanner consists of a ring of detectors that surround a patient. So, what are the parts of the PET scan? The PET scanner consists of a ring of detectors that surround the patient, a computer that processes the data, and the device that injects the radioactive tracer. Working. The radioactive tracer is injected in the body and the patient lies on the scanner bed. The detectors detect the gamma rays released by the tracer, and the computer creates a 3D image of the body based on the detected gamma rays. PET is used to diagnose and study various conditions including cancer, heart disease, and neurological disorders. Now, what is MRI? MRI, its principles. MRI uses a strong magnetic field and radio waves to create detailed images of the body's internal structures. The MRI machine consists of a large magnet, a radio wave transmitter and receiver, and a computer that processes the data. The patient lies on the table that slides into the MRI machine, which creates a strong magnetic field that aligns the protons in the body's tissue. Radio waves are then sent through the body, which causes the protons to emit signals that are detected by the receiver. First, magnetic field is applied. So the body's protons will align in a particular direction. Then radio waves are applied, so which causes the protons to emit signals that are detected by the receiver. Then computer processing is started. Then applications. MRI is used to diagnose and study various conditions including brain and spinal cord disorders, joint and bone problems, and tumors. So fMRI is the another technique. fMRI uses MRI technology to function and measure changes in blood flow in the brain, which can indicate changes in brain activity. The fMRI machine is similar to the MRI machine but includes specialized software to analyze blood flow in the brain. The patient lies on a table that slides into the fMRI machine, which creates a strong magnetic field that aligns the protons in the brain's tissue. The patient performs a specific task such as reading or moving a limb, while the fMRI machine measures changes in blood flow in the brain. The computer processes the data to create a 3D image of the brain that shows areas of activity during the task. fMRI is used to study brain functions and diagnose various neurological and psychiatric conditions. Next is the CT. CT or computer-aided tomography uses X-rays to create detailed images of the body's internal structures. The CT machine consists of a ring of X-ray detectors that surround the patient, a computer that processes the data, and then the X-ray tube that rotates around the patient. The patient lies on a table that slides into the CT machine, which uses X-rays to create a 3D image of the body. The X-ray tube rotates around the patient, while the detectors measure the X-rays and pass through the body. The computer processes the data and to create a detailed image of the body's internal structure. CT is used to diagnose and study various conditions including internal injuries, tumors, and lung disease. In conclusion, each imaging technique has its own strengths and weaknesses and is used for different applications depending on the condition being studied or diagnosed. PET scan, MRI scan, fMRI, and CT are all important tools in the field of medicine and have greatly improved our ability to diagnose and treat various conditions. Fluorescent interference scanning and transmission electron microscope, parts, principal, working, applications, and advantages and disadvantages of each. First, we are going to learn about the fluorescence microscope. First, let's learn about fluorescence microscope. The fluorescence microscope is an optical microscope that uses fluorescence to produce an image. It works by exciting a fluorescent molecule with a specific wavelength of light, causing it to emit light at a longer wavelength. This emitted light is then detected by the microscope and used to produce an image. The main parts of a fluorescence microscope include the excitation light source, fluorescence filters, dichroic mirror, objective lens, eyepiece or camera, and stage. The excitation light source illuminates the sample with a specific wavelength of light, causing the fluorescent molecule in the sample to emit light. The emitted light passes through a series of filters and a dichroic mirror, which separates the emitted light from the excitation light. The emitted light is then focused by the objective lens and detected by the eyepiece or camera. Fluorescence microscopy is commonly used in biology and medical research for studying cellular structures and processes, as well as for diagnosing diseases. So, what are the advantages of fluorescence microscopy? Fluorescence microscopy provides high resolution images of specific structures within a sample, as well as the ability to study dynamic process in living cells, but it has some disadvantages. Microscopy requires to use the fluorescent dyes or proteins, which can be expensive and may affect the function of the sample. Next is the interference microscope. The interference microscope is an optical microscope that uses the interference of light waves to produce an image. It works by splitting a beam of light into two separate parts, allowing the waves to interfere with each other and create an image with high contrast. The main parts of an interference microscope include a light source, beam splitter, mirror, objective lens, eyepiece, and stage. The light source produces a beam of light that is split into two separate parts by the beam splitter. One path passes through the sample and the other path serves as a reference. The two paths are then recombined by a second beam splitter and directed to a specific objective lens, which focuses the light onto the sample. The interference between the two beams of light produces an image with high contrast, which is detected by the eyepiece or camera. Interference microscopy is commonly used in material science and engineering for studying the surface topography and thickness of thin films. Interference microscopy provides high resolution images with high contrast, allowing for the accurate measurement of surface topography and thickness. Interference microscopy can be used for determining the thickness. At least have an accurate measurement of surface topography and thickness. What is the disadvantage? Interference microscopy is limited to studying thin films and cannot be used to study bulk materials. Next is the scanning electron microscope. The scanning electron microscope, or SEM, is a type of electron microscope that uses a focused beam of electrons to produce an image. It works by scanning the surface of a sample with a beam of electrons and detecting the secondary electrons that are emitted from the sample. The main parts of a scanning electron microscope include the electron gun, electron lenses, scanning coils, specimen chamber, detector, and computer. The electron gun produces a beam of electrons which is focused on electron lenses and directed onto the sample. The scanning coils move the beam across the surface of the sample, and the detector detects the secondary electrons that are emitted from the sample. The computer processes the signals from the detector to produce an image. Scanning electron microscopy is commonly used in material science, biology, and other fields of studying the surface topography and morphology and composition of samples. Scanning electron microscopy provides high resolution images of the surface of a sample as well as information about the elemental composition of the sample. And what is the disadvantage? Scanning electron microscopy require a high vacuum which can limit the type of samples that can be studied. Next is the transmission electron microscope. The transmission electron microscope is a resolving power of different microscope. Transmission electron microscope will be dealt later. Next is the resolving power of different microscope. The resolving power of a microscope refers to its ability to distinguish two closely spaced objects as separate entities. The resolving power of microscope is determined by its optical design and the wavelength of light or electrons used to observe the specimen. Optical light microscope. The resolving power of an optical microscope is limited by the diffraction of light waves, and it can typically distinguish between two objects that are about 200 nanometers apart. 200 nanometers apart. Next is the fluorescent microscope. The resolving power of fluorescent microscope is similar to that of an optical microscope, but it can also detect the emission of fluorescence from the specific sample molecule that are labeled with fluorescent dyes. And the electron microscope. The resolving power of an electron microscope is much higher than that of the optical microscope because the wavelength of electrons is much shorter than that of light waves. The resolving power of a typical electron microscope is about 0.2 nm, which allows it to observe the fine details of cells and tissues. Next, scanning probe microscope. The resolving power of a scanning probe microscope depends on the size of the tip used to scan the surface of the specimen. The tip can be small as a few atoms in diameter, which allows the microscope to observe individual atoms and molecules. Overall, the resolving power of microscope is an important factor to consider when choosing a microscope for a particular application. Depending on the level of details required, one might choose a microscope with a higher or lower resolving power. Different fixation and staining techniques for electron microscope. Fixation and staining techniques are essential for preparing biological samples for electron microscopy. There are different techniques available for sample preparation, and each technique has its own advantages and disadvantages. Here we will discuss fixation and staining techniques for electron microscope, as well as the procedures and applications of freeze etch and free fracture methods. Free fracture alla freeze fracture. Fixation techniques. Fixation is the process for preserving biological samples in a stable state, preventing further degradation or changes. Common fixation techniques for EM include chemical fixation. In this technique, samples are immersed in a fixative solution containing aldehydes such as glutaraldehyde or formaldehyde. The fixative cross-links the proteins and lipids of the sample, preserving its structure. This technique is widely used for preserving cells and tissues. Next is the cryofixation. Cryofixation is used for preserving samples in their natural state. In this technique, samples are rapidly frozen using liquid nitrogen or helium, preserving their structure and avoiding artifacts that can occur during chemical fixation. Cryofixation is used for studying protein-protein interactions and other dynamic processes. Staining techniques. Staining is used to enhance contrast and visualize specific components of the sample. Different stains can be used depending on the structure being visualized. Common staining including for EM include negative staining. The negative staining, heavy metal ions such as uranyl acetate are used to stain the background around the sample. This technique is useful for visualizing viruses and large macromolecular structures. Next is the immunostaining. Immunostaining is used to visualize specific proteins or other components in a sample. Antibodies conjugated to heavy metals or fluorescent dyes are used to bind to the target molecules, allowing them to be visualized. Next, freeze etch and freeze fractures technique. Freeze etch and free fracture methods are used to visualize the internal structure of cells and tissues. These techniques involve freezing the sample, then fracturing it to expose its internal structure. In freeze etch, the fractured surface is coated with a thin layer of metal to create a replica of the sample which can be visualized using electron microscope. In free fracture, the fractured surface is directly visualized using electron microscope. Advantages of freeze etch and freeze fracture method. Freeze etch and freeze fracture method have several advantages over other EM techniques. These advantages include preservation of internal structure. freeze etch and freeze fracture methods preserve the internal structure of cells and tissues, providing a more accurate presentation and representation of their structure. No chemical fixation. These techniques do not require chemical fixation, which can reduce artifacts and alter the sample. High resolution. These techniques provide high resolution images of the sample, allowing for the detailed visualization of internal structures. In conclusion, fixation and staining techniques are essential for preparing biological samples for electron microscopy. Freeze etch and freeze fracture methods are powerful techniques for visualizing the internal structure of cells and tissues, and they offer several advantages over other EM techniques. Next is the image processing methods in electron micrograph. Image processing plays a critical role in enhancing and analyzing the images obtained from electron microscope. It involves various techniques to remove noise, enhance contrast, and extract features from the images. Some of the commonly used image processing methods in the electron microscopy are filtering. It is a technique used to remove noise and enhance the contrast of the image. There are different types of filters such as Gaussian filter, median filter, linear filter, etc. Contrast enhancement. This technique is used to improve contrast enhancement. This technique is used to improve the contrast between different parts of the image. First one was the filtering. Next is the contrast enhancement. Some of the commonly used techniques are histogram equalization. Some of the commonly used techniques are histogram equalization, contrast stretching, and Laplacian enhancement. Edge detection. This technique is used to detect edges of the image. It helps in identifying the boundaries between different structures in the image. Some of the commonly used edge detection techniques are Caney edge detection, Sobel edge detection, and Roberts edge detection. Next is the segmentation. It is a technique used to separate different structures in the image. It is a technique used to separate different structures in the image. It helps in identifying different regions of interest in the image. Some of the commonly used segmentation techniques are thresholding, region growing, and active contours. 3D reconstruction. This technique is used to generate 3D models from 2D images obtained from electron micrographs and microscopes. Some of the commonly used 3D reconstruction techniques are back projection, filtered back projection, and maximum likelihood estimation. In addition to the above techniques, there are also various software tools available for image processing in electron microscopy such as ImageJ, Amira, and IMOD. ImageJ, Amira, and IMOD. Overall, image processing methods play a crucial role in extracting information from the images obtained from electron microscopes. This help in enhancing the quality of images, identifying different structures, and generating 3D models, thereby enabling researchers to obtain a better understanding of the biological samples being studied. Which are the major techniques of this image processing? Filtering technique, contrast enhancement, edge detection, segmentation, 3D reconstruction, and estimation with other techniques like available software tools are used. Laser and its parts, working and application, and disadvantages and advantages in biology. Lasers are devices that emit intense narrow beams of monochromatic coherent light through the process of stimulated emission. They have been an important tool in various fields of biology due to their unique properties, including their ability to generate high intensity light of specific wavelengths, precise focusing ability, and low divergence beams. Here is a detailed explanation of the parts, working, application, and advancements of the laser in biology. Active medium, the material responsible for producing the later light. Pumping source, an energy source such as an electric current or another laser caused to excite the active medium. Next is the optical cavity. Pumping source, active medium. Next, optical cavity, a structure that reflects light back and forth through the active medium to produce coherent light. That is something which is called the optical cavity, a structure that reflects light back and forth through the active medium to produce coherent light. Next is the output coupler.
a partially reflective mirror that allows some of the laser light to escape the cavity and form the laser beam. Next is the power supply, provides necessary electrical energy to operate the laser. Working of the laser. The working of a laser is based on the process of stimulated emission. When energy is supplied to an active medium, usually in the form of electrical energy or light, the atoms in the active medium gets excited to move to higher energy level. Active medium excites. So when these excited atoms return to their